Welcome back to my channel where we explore fascinating topics in biology. I thank you all for joining me. Today, we are diving into the amazing world of cell physiology with a special focus on osmosis. Have you ever wondered how water moves in and out of cells, or why a wilted plant can spring back to life after watering? Osmosis holds the answer. Join me as we break down this vital process and uncover its significance in our everyday lives. Let's get started, guys. Let's get started on this important journey of this KCSE question of 1983. Exploring osmosis, a simple experiment for understanding life processes. In this video, I'm just going to start by trying to show you how we can notice that osmosis is taking place by just mere observance of the behavior of plants and water. So let's move. In that photograph, you have a drop of dew on the clover. Have you ever asked yourself how that water gets to that place? Dew formed on the surface of strawberry leaves. A drop of dew on a colocasia leaf. You can look for the meaning of the word colocasia. Introduction. Hello and welcome. I am Mr. Midamo, TK, a retired biology teacher who's still passionate about sharing knowledge. Today, we are diving into the fascinating process of osmosis. Have you ever noticed how soaking a potato in water makes it swell? Or why plants never dry out despite the heat? Osmosis is the answer. And I'm going to show you a simple experiment to demonstrate this vital process. Let's get started. Before we get started, here are the materials we will be using. We will be using a potato. It represents a semi-permeable membrane, sugar to create a hypertonic solution inside the potato, distilled water This is the solvent for osmosis, a beaker to hold the setup, and a scalpel blade for carving the potato so that you have a cavity. So, let me first of all dive on the materials we are going to use in real time. So we will be using potatoes. We have said we use a potato. So here I've shown you the way the potatoes look like, and here I've shown you potatoes of different colors depending on where you come from in this world. There you have the potato which we are going to use. Its scientific name is Soranum tuberosum, tubers exposed to light. So that potato looks like that because it has been exposed to light. We will be using sugar, and there I will show you different types of sugar. Sugar clockwise from top left, white refined sugar. Then we have unrefined and unprocessed cane sugar, which is brown. In this experiment, it's very important that we are going to use distilled water. And here I'm showing you a bottle of distilled water in real Farmacia in Madrid. All right. This one is already distilled water. Remember, they are factory made. We will be using a beaker, and here I've shown you beakers of different sizes to give you a glimpse of how a beaker looks like. We will be using the scalpel blade. Can you see that one here? It's a scalpel blade. Now, among the materials we had been given, so let me go back a bit. Now, this was a question, an essay question of KCSE 1983, and it was question number 5, biology. So, for the purpose of this video, I've identified that the question was testing cell physiology, and you are supposed to write an essay. So, you are provided with the following materials and apparatus: potato, starch powder, distilled water, sugar, tap water, a piece of muslin cloth, a scalpel blade, and a beaker. Out of this list, select the most appropriate ones for carrying out an experiment on osmosis. For each of the ones not selected, give a reason why it is not appropriate. You are being given 3 marks. Draw 2 labeled diagrams, Roman 1, to illustrate how you would set up the selected apparatus and materials. Roman 2, to illustrate the expected results, and C, of what biological significance or importance is osmosis and diffusion. Why is diffusion important? Of what biological significance is diffusion? I was adjusting the camera. And here is what we want to use. So we are not going to use tap water because it contains impurities that might interfere with osmosis. And we want to use starch powder because it's not soluble enough for this demonstration. And we want to use muslin cloth because it is completely permeable. Let's set up the experiment. Before we set the experiment, it's my pleasure to tell you the appropriate materials. Potato acts as a semi-permeable membrane, crucial for the osmosis. Distilled water provides a pure solvent for the process. Sugar creates a hypertonic solution inside the potato. Scalpel blade used to carve the potato for the experiment. Beaker holds the setup. For the sake of clarifying, these are the materials not selected and the reason why they are not appropriate. Starch powder, it does not dissolve adequately to create a hypertonic solution, or it is osmotically inactive. In fact, I'm trying to emphasize the earlier point there. Tap water contains impurities that may interfere with the osmotic process because it has dissolved mineral salts and substances. While muslin cloth is not necessary for this setup, as it does not contribute to the demonstration of osmosis. Again, even if you use it, it is completely permeable. Setting up the experiment. Step 1. Hole out the center of the potato with a scalpel to create a small cavity. 2. Fill the cavity with a concentrated sugar solution. 3. Place the potato in the beaker filled with distilled water. 4. Leave the setup undisturbed for a few hours. By setting up this experiment, we are simulating how osmosis occurs in living cells, guys. The sugar solution creates a hypertonic environment inside the potato, while the distilled water is hypotonic. Let's see what happens over time. Setup diagram. Diagram description. It shows a beaker containing distilled water, a potato with a hollow cavity filled with sugar solution placed in the water, a rebel, potato, sugar solution in the cavity, distilled water in the beaker, and the beaker. There you are. A rebel diagram to illustrate how the selected apparatus and the materials would be set up. Okay? So you can see the diagram. You have the beaker, you have the potato, the scoop or where the space there, distilled water, sugar where you put the sugar crystals, and that is the setup. After a few hours, You notice the cavity is filled with more liquid. What happened? Water from the beaker moved into the potato through osmosis across its semi-permeable membrane. This happens because water moves from an area of low solute concentration, the beaker, to the higher solute concentration, the sugar solution in the potato. This is osmosis in action. Diagram description. Okay, diagram description of the expected results. You are going to show the potato after some time. The cavity in the potato is filled with more liquid due to water entering by osmosis. And then you label increased liquid in the cavity, potato, distilled water in the beaker. Label the diagram Roman 2 to illustrate the expected results. So you can see there is rise in level of water. And you can see the expected results. Those are the expected results. What is the biological importance of osmosis? According to the Kanzo marking scheme, is absorption of water by the root hairs, opening and closing of stomata, turgidity of cells, movement of water in and out of the cell, and finally, osmoregulation. Osmosis is essential for life. In plants, it helps absorb water from the soil, maintaining turgor pressure to keep them upright. In animals, it regulates water balance in cells and tissues. Osmosis works hand in hand with diffusion, which is critical for gas exchange like oxygen moving into your bloodstream. Together, they sustain life as we know it. That diagram or illustration there shows xylem vessels which transport water and minerals from the root upwards. So the water and minerals through the xylem pass through the brew. Can you see the brew? The brew is the xylem. And after that, then you get photosynthesis products which move down through the phloem, the grayish. So, xylem brew carries water from the roots upwards. Phloem, which is orange, carries products of photosynthesis from the place of their origin source to organs where they are needed. Roots, storage organs, flowers, fruits. Note that, e.g., the storage organs may be source and leaves may be sink at the beginning of the growing season. I think I gave you the meaning of the word sink the other time. All right. On the side of the animals, I'm pleased to talk about the red blood cells and osmosis. I've cleared with the plants. So you can see the effect of osmotic pressure on brand cells if they are put in a hypotonic solution, isotonic solution, and hypotonic solution, which are those ones have already been defined in my other biology videos in cell physiology. So have a point of checking my channel if you are observing it for the first time or watching the video for the first time to check cell physiology where I've defined those ones. But simply, hypertonic means a concentrated solution. Isotonic means the solution is having the same concentration as the concentration of the red blood cell, and hypotonic means the solution is dilute. Solution is dilute. Typical mammalian red blood cells. Now you can see, I will explain what is in A, what is in B, what is in C, and what is in D below. So, A are the cells as seen from surface. B, they are in profile, forming luliacs, which is a phenomenon in which red blood cells stick together and form chains or stacks that resemble a stack of coins. That is what we mean by luliacs. C, the cells are rendered spherical by water. D, rendered crenate or shrunken and spiky by salt. All right. C and D do not normally occur in the body. The last two shapes are due to water being transported into and out of the cells by osmosis. Biological importance of diffusion. Because osmosis is a special type of diffusion. So what is the biological importance of generally diffusion? Movement of mineral salts in root hairs, translocation of food substances, gaseous exchange in animals, and excretion. So in general, osmosis is important because it facilitates the uptake of water by plant roots. It means cell turgor in plants, preventing wilting, and it regulates water balance in animals, preventing bursting or shrinking. Importance of diffusion, because osmosis is a special type of diffusion, is it enables gas exchange during respiration, e.g. oxygen into cells, carbon dioxide out. It aids nutrient absorption across cell membranes. And 3, facilitates the elimination of waste products such as urea in organisms. So osmosis, if I dwell on it a bit, is the spontaneous net movement or diffusion of solvent molecules through a selectively permeable membrane from a region of high water potential, region of lower solute concentration, to a region of low water potential, region of higher solute concentration, in the direction that tends to equalize the solute concentration on the two sides. Perfect definition, my dear people. On the other hand, diffusion is the net movement of anything, for example, atoms, ions, molecules, or energy, generally from a region of higher concentration. Concentration is a measure of how much solute is present in a solution, and it can be expressed in various units such as molarity, weight percent, and mole fraction, to a region of lower concentration. Diffusion is driven by gradient in Gibbs free energy. And Gibbs free energy, it's a measure of the useful energy available to do work, taking into account both the system's internal energy and the entropy or disorder associated with that energy. That's what we mean by Gibbs free energy. Or chemical potential. So diffusion can be either driven by Gibbs free energy or chemical potential, and the chemical potential describes the tendency of a substance to move or react with a system, guiding processes towards equilibrium. Guys, give a comment on that definition. Is it good? Is it clear? Do you like it? Are you smiling when you read it? Conclusion. So, that's osmosis, a process that keeps plants green, cells hydrated, and life thriving. If you enjoyed this experiment and want to explore more biological concepts, hit the like button and subscribe for more content. Let me know in the comments what experiment you would like to see next. Thanks for watching, and remember, science is all around us. So guys, after that, I've given attribution or acknowledgement to all the owners of all those photographs or any diagram which was there. So from here, I'll end the video there. You can go over this by yourself, or if you want to do more research, you can go to Wikipedia and find more about those photographs I've given. So I've got all these attributions. Those are for the potatoes. Then we have the scalpel bread. All right. We have the sugar. We have the distilled water. All right. We have the xylem. All right. We have the xylem. Okay. All right, we have red blood cells. All right. We have the red blood cells again there. And